Can somebody confirm that you're here? All right, no problem. If there's no questions, let's begin. I think we're behind schedule. Let's see. We are supposed to be starting chapter 8 today, but we're still in chapter 6. Hopefully we'll get started on chapter 8 today. All right, last time we had talked about the effect of oxygen on microbial growth. When we're talking about culture media, you should realize that the word medium with an M is singular and plural for more than one is media. It is a liquid or a solid solution containing all the nutrients needed for microbial growth. Generally, when you get them, if you were to come into the Clark lab, it would be sterile, and that means it has no living microbes in it. It is sterilized by putting in an autoclave, and we'll talk about that in a little bit. The inoculum is the microbes you introduce into the media. We call it a culture, and that is the microbes growing in a culture medium. There are a wide variety of media available in microbiology. If you were coming into the lab, I would hold up a book that each page has a page discussing one media, and the book is about that thick. So that tells you how many different media we have in microbiology. The media is to support microbial growth. It must provide an energy source, a carbon source, sources of nitrogen, sulfur and phosphorus, as well as trace elements. And if the organism needs them, organic growth factors. Now, if the organism doesn't need a carbon source or a nitrogen source, you do not need to supply them. Some auxotrophic, what's the word I'm trying to say? Photosynthetic organisms, which are autotrophs, can supply their own carbon source, pulling carbon down from the air, as well as there's a few that can pull down nitrogen from the air, but everything else would need to be supplied. Any questions about any of that? Like for humans, we have to have an energy source, a carbon source, a nitrogen source, a sulfur source, a phosphorus source, trace element source, and a source of organic growth factors like vitamin C. Agar is a complex polysaccharide derived from seaweed. We use it as a solidifying agent for a culture media. And so you put agar into the liquid media and it'll make it solid. Agar is a substance similar to Jello. Agar is usually in a test tube or a petri dish, a petri plate. The tubes can be of two types, slants and deeps. And let me get a picture so I can show you. So the tube being here, and pretend I'm a good artist. A deep is one where the agar goes across, and a slant is where the agar is slanted. Any question about that? So this is a slant, that is a deep. Agar is not generally metabolized by microbes. You don't need to know the properties of agar. We have different types of media. There's a chemically defined media, and this media has the exact chemical composition known because every molecule in the media is one that we add. You may need to add many growth factors, and if you do need to add many, then we call that organism fastidious. It does not grow easily. The chemically defined media is normally only used for growing autotrophs because they're, well, easier to grow. They supply a lot of their reagents. Let me blow this up a little bit so we can see it easily. So this is a chemically defined media. When we add distilled water, you're not adding any other molecules other than H2O, and anything dissolved in the water, like carbon dioxide, will be dissolved in the water. And then we add potassium phosphate, magnesium sulfate, sodium chloride, ammonium phosphate, and that's to add nitrogen as well as phosphate. And then glucose. So glucose is both the carbon source and the energy source. And that's it. We know everything that's in that media. And that's the chemically defined media. Any questions about that? Most of the organisms that we grow in the lab would not grow on this media because they need other additives. And what we do in the lab is generally give students a complex media. It contains extracts and digests of yeast, meat, or plants. And these digests provide vitamins and organic growth factors. The exact chemical composition of a complex media is not known because, like for this media here, which is a chemically complex media, we're adding beef extract. All of the molecules that were in the cow that would be ground up to make the beef extract will be put into this complex media. And we have no idea what all of those molecules would be. It will obviously depend on what the cow has been eating. Any questions about that? All right, so a solid media would have a peptone, that's a partially digested protein, beef extract, sodium chloride, agar, and then of course water. And that's a complex media. The peptone, as well as the beef extract, supply the carbon, the nitrogen, and the sulfur sources. And complex media is used for heterotrophs, bacteria and fungi that require an organic carbon source, like E. coli, for example. Any questions about any of that? We have discussed some complex media, and we'll talk about them a little bit later, but nutrient agar with yeast extract, and tryptic soy agar, or TSA, are three complex media that we commonly use in the lab. There are other types of media, like an anaerobic media. This media is important for growing obligate anaerobes. They must avoid exposure to oxygen. An anaerobic media has a reducing agent in it, that is a chemical such as sodium thioglycolate, that removes the oxygen from the media. So sodium thioglycolate, at least at the bottom of the tube, will be oxygen depleted. And then the anaerobes can grow in the bottom of this tube. What you do to remove the oxygen from the media after it's first made, because obviously it's the oxygen will be dissolved in the water, and when you stir it in, it'll be pulled into the media. You can autoclave it, and that drives off the absorbed oxygen. There are also selective media. These are media that will halt or inhibit the growth of a particular group of microorganisms. Other organisms may grow in the media. An example is 7% sodium chloride media. It is selective for organisms that can grow in 7% sodium chloride. Most organisms do not grow above 2% sodium chloride. And so only halophiles, meaning salt lovers, can grow in this media. Any question about any of that? All right. There are also differential media. These media provide differences between different species growing on the media. The differences are not related to how well the organism can grow on the media. It may just distinguish the colonies. They may have different characteristics or different colors when they grow on the media. An example of a differential media is blood agar, and that allows us to distinguish between the different types of hemolytic activity that the organism has. There is no hemolysis where the blood of the agar is not lysed from the media, come on mouse, all the way up until we see the growth. So this white area here is the growth of bacteria. The blood is red all the way to the growth. And this we call gamma hemolysis or no hemolysis. There's also partial hemolysis where some of the red blood cells are hemolyzed, but not all of the red blood cells are hemolyzed. And so you have the red, and then you have a dark, ring around the off-white growth. That dark ring, which usually looks greenish or darkish, is from partial lysis of the red blood cells, but it's not complete lysis. Why? It's got color to it. But there's partial, so it's not red like this area. And it's most visible actually inside the alpha of the bacteria, which is sort of a off-white color. This is called partial hemolysis or alpha hemolysis. There's also beta hemolysis, and that's complete hemolysis. Here we have the white B, which is the bacterial growth, and it secretes a hemolysin, a protein that lyses the red blood cells. And so we have the red, come on mouse. Sorry, my mouse keeps dying. Right here. And then when we come up here, it's no longer red. And then right there, you see how it's clear? You can call that white, but in reality, that area is clear. Oops, didn't mean to do that. That's from the complete hemolysis of the red blood cells. This we call beta hemolysis or partial hemolysis. Excuse me, beta hemolysis or complete hemolysis. I'm trying to get my mouse to work. You notice this dark ring around the beta hemolysis? This is alpha hemolysis. It is normal that you see a ring of alpha hemolysis around beta hemolysis. Any question about that? Anyways, that's blood agar. Another type of media is selective and differential media. It is both selective and differential. EMB or eosine methylene blue media is both selective and differential. It's selective against gram-positive cells. They don't like eosine and methylene blue getting in their cell wall, and it prevents the bacteria from growing. And then it's differential on the basis of lactose fermentation. When you get lactose, there's lactose in the media. When you get lactose fermenting, it causes the bacteria to look dark in the colonies here, or greenish, which it looks greenish there. And if it's non-lactose fermenting, the colonies will look whitish. Any question about that? So only the gram negatives will grow on this. And if it is differential, it will ferment the sugar lactose and look black or greenish. There's also an enrichment media. It encourages the growth of certain bacteria. So it is therefore selective, but we use it to encourage the growth of something. It increases the numbers of desired microbes to detectable levels. It's particularly useful if your target microorganism is only a small proportion of the total population. So we use an enrichment media when we're looking for bacteria in the soil or some other environmental source, or like a fecal sample when we're looking for a pathogen in a patient. And the pathogen would be low in number in the feces because the feces have so many other bacteria in them. An example, you take sampling of bacteria from the soil, and there's less than 1 in 1,000 of the bacteria of interest in a culture of bacteria from the soil. We can put it in an enrichment media, like if we're looking for phenol metabolizing bacteria, you make an enrichment media where, for example, phenol would be the only carbon source. And so the only thing that will grow in here besides an autotroph is the phenol metabolizing bacteria, because phenol is the only carbon source. And then you grow the bacteria in that. And if needed, you can take the bacteria which are enriched in that tube and then put it into another tube and then further enrich it. And you need to have the bacteria up at least to 1 in 100, because when you streak it out on a Petri dish, you're likely to only get about 10 colonies. And so if you do 10 plates, in reality, you'd have about 1 colony of interest. Now that's not very good, 1 out of 100. It'd be best to get it up into 1 out of 10. And that's why you can enrich in more than 1 tube. You enrich in 1 tube, take that enriched growth, put it into another tube, and enrich further. Any questions about enrichment media? All right, this table is showing you the different types of media that I've already discussed. Let me mention that a pure culture contains only 1 species in it. Louis Pasteur came up with the concept of working with a pure culture. An easy way of getting a pure culture is, of course, to streak it out on an agar plate and get a colony, and then take from that colony and streak it out on another agar plate and get a second colony, so that you've gone through 2 colony isolation events. And then grab from that second colony, and then the chance that that will be a pure culture will be very high. It may not be a pure culture the first time you do it, because the colony could be formed from more than 1 cell, like a clump of cells or 2 cells. And therefore, when we count colonies on a plate, we don't say how many bacteria we have in the sample. We say how many colony forming units we have. Anyways, to obtain single colonies is the critical part of obtaining pure cultures. And what you do is you streak it out for isolation 2 times. And even then, you're not guaranteed you'll have a pure culture, but it'll be greater than 95% pure. Okay? If you only restrict it out about 1 time, it'll be about 75% of pure culture. Any questions about any of that? This is showing you how to streak it out for colony isolation. Streak it out, get your bacteria on a loop, streak it out in quadrant 1. And then set up quadrant 2, sterilize your loop, start streaking on quadrant 2, and come into quadrant 1 where it's been diluted, meaning the highest concentration of the bacteria over here, the more dilute bacteria over here. You want to bring it in 3 to 4 times in quadrant 1, and this is over that. But you want to do it about 3 to 4 times. And then streak out quadrant 2, and then sterilize your loop and bring it in quadrant 3, and then bring it into where it's diluted in quadrant 2. And it's showing you only 1 time. In reality, you want to come back 3 or 4 times in the diluted portion of quadrant 2. Any questions about any of that? And then the goal is someplace along the line, it might be here, it could be here, it could be here. You will dilute out the bacteria enough that you will be streaking out single colony forming units, which will then go on to form a colony, which are isolated, not touching other colonies, such as those right there. And as you can see, we had a mixed culture here. Blow that up a little, because there are definitely 2 colony forming units here, a red colony and a yellow colony. The red ones we got a pure culture from, but the yellow ones, they're not isolated. Like if we were to grab from this colony, we might get red cells out of there. So you need to streak it out again. Let's make sure I do that right. Colony streaking only works when the organism of interest is large enough in proportion to the total microbial population. Like here, the yellow colonies are not high enough in the population to get a pure culture from doing 1 streak. Any question about any of that? Come on, mouse. All right, so let's talk about microbial growth a little bit. Remember, microbial growth refers to the increase in number of cells, not an increase in the cell size. Bacteria grow by 3 means. Most bacteria grow by binary fission, where 1 cell splits into 2. It literally divides down the middle. We'll see that. Bacteria can also grow by budding, where 1 parent cell puts out a bud. And then there's a few bacteria that reproduces by bacteria reproductive spores. Those usually have tips at the filament bacteria, which has a tip, and they'll have a few reproductive spores on it. This picture is showing you binary fission, where 1 cell begins dividing. It replicates its DNA, and then the DNA moves towards opposite ends of the cell. And then the cell literally starts dividing in 2, growing the cell wall and the cell membrane right between the cells. And then at this point, they have the cell wall and the cell membrane grown in between them, and then the cells split into 2. And here's an actual electron microscopic picture of a cell, which is going through binary division, binary fission. Any questions about that? Bacteria can also bud where the parent cell puts out a little bud, which then grows and gets larger. And eventually, when this cell is close to the size of the parent cell, the two will separate. Let's watch a little video on budding. Oh, there it is. So watch that one. See how it's putting out a bud right here. And look at that. That's the DNA. Oops, come on. The DNA is dividing, and there the DNA is actually moving into the bud. And when this cell gets to be about the same size as the parent, a little smaller, it then forms a wall, cell wall in between them. And now that cell is complete. And the parent cell is putting out another bud, and so is the newly budded cell. We'll word it that way. Any questions about that? I'm going to take a 30-second break. I need to turn off something. Sorry about that. The growth of bacteria cultures, we use the generation time. For some reason, students often have trouble with the generation time. The generation time is the time required for a cell to divide or for a population to double in numbers. The two are the same. It varies considerably between species what the generation time is. It can be as fast as 20 minutes per generation, meaning E. coli, which is one of the fastest cells to double, can go from 1 cells to 2 cells in 20 minutes, or a population of 1,000 E. coli can double to 2,000. Any question about that? Most bacteria take longer than that, and most take about 1 to 3 hours to double. The slowest that we know of is Microbacterium leprae, which takes 10 days to double. The Mycobacterium family have mycolic acid in their cell wall, and that's a very complex molecule which the cell needs to make before the cell can double. And so that's why the Mycobacterium tend to be slower growing than most other bacteria. Any question about any of that? What students have trouble with the doubling time is, I guess we're used to talking about like 60 miles per hour. The doubling time, when we say 20 minutes, it's 20 minutes per generation. That per generation is understood. And so this is a time per generation. It is not a number of generations per time, like 60 miles per hour. That's a unit per hour. And that is not the generation time. Is that clear? All right, I don't hear anything, so let's move on. Microbiologists like to graph the generation time on a semi-log plot. So you put the log of the number of cells on the y-axis, and then the x-axis is just the number of generations. So this is a semi-log plot because the x-axis is, I guess, a linear numerical numbering system. If we were to have it numerical numbering system on the y-axis, and we were to plot the growth would look like this, exponential. And there would be 1 cell dividing into 2, 2 into 4, 4 into 16, 16 into 32. There would be a whole bunch of points down here. which aren't going to get separated from the graph. And then our next point right here will be on the ceiling. And that's why we don't like to use a linear plot. Another reason is that when we use the logarithmic plot, we get a very nice equation for explaining the growth. It's simply a linear equation, y equals mx plus b. This equation is something like y equals mx squared plus nx plus b, a much more difficult equation to work with. Another reason is that when we use the log plot, we scatter these points down here, so they're not all bunched together. And then the next point here will be about right here. So it's still on this paper. It's not on the ceiling, whereas the next point here will be on the ceiling. Any question about any of that? Anyways, that's why we like using semi-log plots. It scatters the initial points. It allows you to bring in one more point on the graph here. And it's a much easier equation to work with. When we're talking about growth, it's important to realize that bacterial growth have different phases of growth. And this is generally true for almost all living populations, this growth curve. It can be generated experimentally, like when you put something growing in a liquid media and then you put it on a solid media, or vice versa. You normally have 4 growth phases where the number of bacteria cells are on the y-axis and the time is on the x-axis. Initially, when you take the cells out of one media and put them into another, there's going to be a lag phase. Why do we have a lag phase? Because the organisms are not used to growing in the new media, and they have to make the enzymes they need for growing in the new media. Any question about any of that? Once the organism then has the enzymes they need for growing in the media, they then start growing exponentially. And that we call the log growth or the exponential growth phase. So they grow very rapidly. That starts to bend off up here. And why that happens is, why things are growing quickly is because there's lots of resources around, lots of nutrients, very few wastes down here, lots of space for the organisms. Well, up here, the bacteria numbers are increasing. There's fewer nutrients around, there's less space, and there's more waste products around. And that will slow down the bacterial growth. Eventually, the growth will slow to the point where the population numbers do not increase. They don't decrease either. And we call that the stationary phase of the population. In the stationary phase, the number of new cells equals the number of dying cells. So the population number stays about the same. Why this is happening is because the lucky cells have a little bit of space to divide, a little bit of nutrients, and they don't have too many wastes around. So they're dividing. The unlucky cells do not have those things, and they're dying. Any question about any of that? All right. After the stationary phase, we then enter the death or decline phase. This is when the number of cells dying is greater than the number of cells dividing. Early on in the death decline, you do have some cells, not very many, but some cells that are dividing. Later on in the death or decline phase, you have no cells which are dividing. They're only dying or maybe staying stationary. And then this will go until the population reaches 0. That can be, this can be on the order of days or weeks. And if it's endospores, it can be millennia or centuries before all of the cells die. So it will depend on the characteristics of the bacteria, how long this will be. All right, any questions about the phases of growth? If not, the next slides are all in the in-class assignment, the discussion. And so it was talking about the measurements of microbial growth. And I'm going to skip that because we're behind schedule. And you got that from the in-class assignment. Any questions about any of that? All right, if there's no questions, let's move on to chapter 8. As always, I give you a first slide, which gives you the major goals and the rough lesson outline. Always know the terms. We'll talk about each of these and be able to describe replication, transcription, and translation. Also understand the different types of mutation. Any question about what we're doing? We're first going to talk about the structure and function of the genetic material. You should know that the genetic information has a flow, and we'll talk about that, how the DNA or the hereditary material flows from one cell to another. We'll talk about DNA replication, RNA transcription, and protein translation. We'll then move on to the regulation of bacterial gene expression. We'll talk about the operons, what they are, how genes are expressed, how they're turned on and turned off. We'll talk about mutation, and then we'll talk about genetic transfer and recombination. And then lastly, we'll end with genes and evolution. Any questions? All right. Genetics is the study of what genes are, how they carry information, and how that information is expressed, as well as how genes are replicated. A gene is a segment or a piece of DNA that encodes a functional product. The product that it encodes is usually a protein, but it can be an RNA. Here we're looking at a gene on a chromosome. So this region of the DNA is gene 1. It encodes for a product. And this is the region coding for gene 2. You'll notice in this example, there's some DNA in between the DNA coding for gene 1 and gene 2. And this is showing you a chromosome, and there's different locations for the genes, which are illustrated here. Any question about any of that? The genome is all of the genetic material in the cell. The study of genomes is called genomics. The genotypes are the genes of an organism. For example, blue-eyed people have the genotype, the genes of the organism, lowercase b, lowercase b. They have to have that in order to have blue eyes. The phenotype is the expression of these genes. So the expression of the lowercase b genotype is blue eyes. Now for brown eyes, individuals can have the genotype uppercase B for brown or lowercase b for blue. Lowercase is recessive, so the phenotype in this case is brown. But brown-eyed individuals may also have the genotype uppercase B, or meaning brown-eyed individuals, when that's their phenotype, they have brown eyes, their genotype can be uppercase B, lowercase b, or two uppercase B genes. Any question about that? The phenotype is encoded by the genotype. Here we're seeing a chromosome for E. coli. And on this chromosome are the genes. E. coli normally has only one chromosome. On the chromosome is both DNA and proteins. I think we've talked a little bit about histone proteins, which you only find in, well, I shouldn't say only find, you find them in eukaryotes. You do not find histone protein in the domain bacteria. The DNA is a polymer of the nucleotides. You do need to know that DNA is a polymer of 4 nucleotides, but you can learn them as adenine, thymine, cytosine, and guanine, or as A, T, C, and G, which is shown here. DNA in our cells is normally a double helix associated with proteins. Proteins aren't shown here. And that double helix has the sugar phosphate being the backbone of the DNA, meaning the sugar phosphate sugar holds the DNA together, at least in one strand of DNA. The nucleotides are on the steps of this double helix, and the nucleotides hydrogen bond with the other nucleotide on the second or corollary strand of DNA. And it's only the hydrogen bonds holding the 2 strands of DNA together. There are no covalent bonds holding 2 strands of DNA together. On the other hand, a linear strand of DNA is held together by covalent bonds in the sugar phosphate sugar. Any questions about any of that? You should know that the 2 strands of DNA are anti-parallel, meaning one strand goes this way and the other strand, if I can show it, we'll go like that way. If you were a chemist, I'd say this is the 5' end of this one, that's the 3' end of this one, that's the 3' end of that one, and that's the 5' end of this one. And that is just referring to the carbon in the sugar. That's the 5' carbon, and this is the 3' carbon. And you don't need to know the 3' and the 5', just that they're anti-parallel. The flow of information can happen in different ways. When we're talking about the flow of information within a cell, this is called the vertical flow of information. And it's also known as the central dogma, that the information flows from DNA to RNA in transcription to protein in translation. And in our cells, from our DNA, this is the only way the information flows. The information that I know of never flows from protein to RNA. And in our genes, it never flows from RNA to DNA. We will talk about later in the term where there's a virus where the genetic information does flow from RNA to DNA. But outside of that one family of viruses, retroviruses, that is not seen, where information flows from RNA to DNA. And that's why this is known as the central dogma. The information always flows this way, at least in our genes. When we're talking about the flow of genetic information, it's important to realize that information can flow within a cell. Information can flow between generations of cells, or we could say between different generations, like a parent and an offspring. Or the information can flow between cells of the same generation. Within a cell, the DNA, the information in DNA flows from DNA to RNA in transcription to protein in translation. Between generations of cells, the DNA flows from the parent cell, which divides, and the DNA goes to one side of the cell and the other DNA to the other side of the cell. And then the cell divides into two so that each daughter cell gets an exact copy of the parent DNA. Barring mutation, it's an exact copy. That's the flow of information between cells. Now, between generations, the flow of DNA flows from the parents, and humans have two, to the offspring, which is usually one, but it could be more. Any question about that? The flow of information between cells of the same generation has the information flow out of one cell and into another cell that we call a recombinant cell. Usually the DNA comes out of this cell, like when this cell dies, and then there's free DNA, and then the free DNA is incorporated by this recombinant cell, so that this cell gains DNA from another cell, which would be this cell. And please note, I've broken and changed this graph to make it a little clearer that DNA from here doesn't recombine to there, because if you knew anything about genetic recombination, that's not what they're talking about. They're talking about the DNA coming out of this cell and then going into that cell. Any question about any of that? The vertical flow of information within a cell, where DNA, the information in DNA flows to RNA to protein, has different languages as well as different letters in the alphabet. The DNA, the information in DNA has the language of DNA nucleotides, and there are 4 of them. The language in RNA is also in nucleotides, although it's RNA nucleotides. And there are also 4 letters in the alphabet. The information in protein, the language is amino acids, and generally speaking, most organisms have about 20 letters or 20 different amino acids in a protein. To compare that with English, we have 26 letters in our alphabet. One way of saying this is the information in DNA would be something like, that is a dog, the original information. The information in RNA would be a different dialect, but essentially the same. That is a dog. And then the information in the protein would be a totally different language. Quello è un cane. The same information, just in a different language. All right, let's talk about the flow of genetic information between generations of cells, where the information flows from a parent cell to 2 daughter cells. And this is called the vertical flow of information. What happens is the DNA replicates, and when DNA replicates, it is semi-conservative. Let me blow this up. What we do is we separate the 2 strands of DNA and then use one strand as a template to make the new strand. So here, the enzyme, which is polymerase, let me see, there it is, sees a G here, so it's going to put on the C, which is the complementary base to G, meaning C pairs with G. And then it'll C move down to the next nucleotide on this strand. Sees a C here, so it'll put on a G nucleotide, because that's the nucleotide that pairs with the C. And then move down, sees an A, it'll put on a T. This we call the semi-conservative model of replication, because one strand is used as a template to make the new strand. And then the daughter cell, one daughter cell, will get one strand of the old DNA and one strand of newly replicated DNA. Semi-conservative model of DNA replication. Any question about any of that? So DNA replication is semi-conservative, the black strands being the original strands, and then the daughter cell will get one original strand and one newly replicated strand, and the other daughter cell the same. This is called the semi-conservative model of DNA replication. The conservative model of replication was that one daughter strand would get both of the original strands of DNA from the parent cell, and the other daughter strand would get two newly replicated strands. The liberal model, I have no idea what that was, okay? All right, any questions about any of that? This is showing you how DNA is put on. This is the template. The RNA polymerase, or excuse me, the DNA polymerase sees the adenine here, and so it'll put on a thymine. What you do is you get a triphosphate, and then you split off two of the phosphates, and then have that phosphate bind to the ribose. And so two pyrophosphates are released. Another thing I need to mention is that the replication of DNA happens with three enzymes. It'll be discussed in the video. Let's go ahead and start that, and I'll do my little talk. You need a helicase to open up the two strands of DNA and make the strands single. You need a DNA polymerase, which reads one template of DNA and puts on the correct nucleotides for the newly replicated strand. And then finally, you need ligase to stitch or put the strand of DNA together. So those three enzymes are the important ones. Boy, that still hasn't loaded. I might need to refresh this. Yeah, I needed to refresh it.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All right, any questions about the video?
Bounty versus the old family dish towel. Drying with a fresh sheet.
So DNA replication is continuous on the leading strand because the DNA polymerase moves this way on that strand, and the helicase is moving this way. There's the helicase right there, unwinding the DNA. On the lagging strand, the DNA polymerase has to go this way, and yet the helicase is going that way. So what you do is you build this one in short segments called Okazaki fragments. And it requires, obviously, many Okazaki fragments. What else do I want to say? We talked about the helicase opening up the two strands of DNA, making it single-stranded, so the DNA polymerase can come on and put in the correct nucleotides of the newly replicated strand of DNA using the template, the old strand of DNA. And then the ligase seals the DNA together. Whenever you put on a new piece of DNA, so let's say we're starting right here, and then this is going to go on, this new stretch of DNA isn't covalently linked with this stretch of DNA. And you need to ligate them together with the enzyme ligase. In the leading strand, there will only be two places where ligase needs to work, wherever the DNA polymerase starts and wherever it ends. But on the lagging strand, you need to put it at each side of the Okazaki fragment, meaning you need to ligate that Okazaki fragment to this stretch of DNA and this Okazaki fragment to that Okazaki fragment, etc. So the ligase will be working quite a bit on the lagging strand of DNA. Any questions about any of that? Like I said, you should know the three major enzymes: helicase, which opens up the two strands of DNA, DNA polymerase, which makes the newly replicated strand of DNA using the template, the old strand of DNA, as its template. And then the DNA ligase. Those are the three main enzymes. Any questions about that? So DNA is only made in the 5' to 3' direction. It is started with an RNA primer, and I'm not really going to test you on that. The leading strand is synthesized continuously, the lagging strand discontinuously in Okazaki fragments. And all of the fragments of DNA, including on the leading strand, are joined by DNA ligase. All right. That's it for DNA replication. Any questions about that? If not, let's move on to transcription. Transcription is the synthesis of RNA from DNA. DNA is transcribed to make RNA, and you can transcribe mRNA or messenger RNA, tRNA or transfer RNA, or rRNA or ribosomal RNA. When the DNA, not the helicase, DNA, so I guess it'd be the RNA polymerase, is making the RNA, how does it know which RNA to make? How does the polymerase know to make messenger RNA or tRNA or rRNA? Anyone know? This is simple genetics, but I know you guys haven't had genetics. That's why I'm asking. All right, well, think about that and remind me to answer it, because it's not really that difficult. You just need to know. Transcription begins when RNA polymerase binds to the promoter. The promoter is the first region of a gene, and it actually encodes for the RNA polymerase to bind here. And then transcription proceeds in the 5' to 3' direction. So RNA is made in the 5' to 3' direction. And then the RNA polymerase moves down the DNA, transcribing or making RNA. And transcription stops when the RNA polymerase reaches the terminator. The terminator is a region of the gene or a region of the DNA that codes for the RNA polymerase to fall off. And once it falls off, transcription stops. Anyone have an answer for how does the RNA polymerase know which RNA type to make? When does RNA polymerase make messenger RNA? Or when does it make tRNA? All right, think about it. It's important to realize that when DNA is replicated, all of the DNA is replicated. When the DNA is transcribed, only some of the DNA is transcribed. Only the genes are transcribed. Any information or any DNA that is not a gene will not be transcribed. So the non-coding DNA is not transcribed. Now in bacteria, most of the DNA codes for something, meaning most of the DNA is genetic information. And oftentimes you'll have one gene, let's see if I can show that, one gene right next to the other gene. However, in eukaryotes, you don't have that. You might have one gene, I can't even show it. Why am I not? There it is. Just have to get it in the right angle. One gene may be over here, and the other gene may be way over here. And in between, we have non-coding DNA. The non-coding DNA is never transcribed. Okay? What the non-coding DNA does is not fully understood, but in eukaryotes, it is known that the DNA coils up to form the chromosomes. The non-coding DNA probably does code for the coiling up of the DNA to form the chromosome. So it's probably doing that, and it probably has some other functions as well. I already mentioned about the promoter. The promoter isn't strictly speaking part of the gene that codes for something. Although I wouldn't call it non-coding DNA, it has a different function. It's coding for RNA polymerase bind here. Anyone have an answer for when does the DNA transcribe, make RNA, messenger RNA versus tRNA? All right, think about that. Here we're looking at a picture of RNA transcription. This is an artist's rendition, DNA transcribed into messenger RNA, transcribed into protein. And there is an actual, it's not an electron microscopic image. I think this is a fluorescent image. DNA and the RNA polymerase, and there's the RNA right there being made. As I mentioned, the DNA will be transcribed either into messenger RNA, tRNA, or rRNA. If the DNA is transcribed into messenger RNA, it'll be translated into protein. If the DNA is transcribed as rRNA or tRNA, that's it. There is no protein translation. Only some genes are transcribed as rRNA or tRNA. Most genes are transcribed as messenger RNA. And if you knew my last question, you would know why. But you can think about why is one gene transcribed as messenger RNA and another gene transcribed as tRNA. All right, so this is showing you protein transcription. And I think we're going to stop here. And I'll continue here next time, because we need time to do the lab today. Today is Tuesday, isn't it? Yes. So I'll see you at 6:30.
Quick question. I'm so sorry. I have been drastically doing my unknown lab crazy bad and not getting you the tests requests correctly. And so I finally responded to your request again, because I initially sent it via email and then it was supposed to be via that page, and then I forgot to say what number I was. Lo and behold, I took tomorrow off so that I can hopefully like powerhouse this essay out essentially, but I haven't even done anything on it because I've been unfortunately doing all of this wrong.
Okay, let's talk after the lab about this, because this will be a while, but
Okay.
You're not that far behind. As long as you get started soon,
Okay.
you can still finish in time. So you're
Okay.
not in any trouble.
Oh God, thank you. Okay, I am totally okay talking after the lab. Thank you so much. I appreciate you taking some time after that. I'll let you go now. Thank you.
All right.